Good morning, everyone. It's just a pleasure to welcome you all this morning to Pediatric Discovery Day, which for me is almost like Christmas Day itself, except I'm pretty sure that the boxes under the tree will not contain the keys to a Porsche, which I really, really would like. But nonetheless, this occasion gives us the opportunity to celebrate the pursuit of knowledge and innovation in child health research. Today, we have the distinct honor of hosting an outstanding physician scientist, an accomplished leader in pediatric infectious diseases, and a dedicated mentor for the next generation of scientists and physician scientists, Dr. Toni Darville. Dr. Darville is Distinguished Professor of Pediatrics and Microbiology and Immunology at the University of North Carolina at Chapel Hill. She serves as the Director of Pediatric Infectious Diseases, the Vice Chair for Pediatric Research, and the Scientific Director of the Children's Research Institute at UNC Children's Hospital. In addition, she is co-director of the UNC MD PhD program, where she helps guide the development of physician scientists across discipline. And it's not surprising when you add up her effort allocation, it equates to 140%. And we're going to hear a little bit about that this afternoon, how one is able to accomplish all these important responsibilities. Dr. Darville is a physician scientist, and she's devoted her career to an understanding of the pathogenesis of Chlamydia trachomatis, an incredibly important bacterial sexually transmitted infection that really has a worldwide range. Her work spans basic immunology, translational studies, and clinical investigation, with the overarching goal of defining host responses that drive disease and then leveraging those insights to develop effective vaccines. Through elegant experimental models and human cohort studies, her laboratory has helped define T-cell mediated immunity, host genetic susceptibility, and inflammatory pathways that underlie reproductive tract disease and infertility. Dr. Darville is an enormously productive scholar. She's authored more than 250 peer-reviewed publications, reviews, and book chapters. She has maintained continuous NIH support for over 30 years. Her leadership extends beyond the laboratory. She is past president of the Chlamydia Basic Research Society, a recipient of the Pediatric Infectious Disease Society Distinguished Research Award, and an internationally recognized authority on chlamydial infections and vaccine development. Dr. Darville received her BA degree from the University of Arkansas and her MD degree from the University of Arkansas for Medical Sciences. She also completed her pediatrics residency and fellowship in pediatric infectious diseases at the University of Arkansas for Medical Sciences, where she began her academic career before joining, yes, the University of Pittsburgh and UPMC Children's Hospital. Indeed, where she served as professor and held the Carol Ann Cromer Endowed Chair for Pediatric Research. She later moved to the University of North Carolina, where she now leads a vibrant and highly collaborative research group. Now, I have to say, equally important to all of Dr. Darville's many research contributions is her commitment to mentorship and training. She has guided numerous fellows, graduate students, and junior faculty, and through her leadership of the MD PhD program, as well as the Children's Research Institute, she continues to shape the future of pediatric physician scientist education and training. She also remains an active clinician caring for children with complex infectious diseases, all the while maintaining a terrific research laboratory team. This morning, Dr. Darville is going to share insights from her work on immunity and vaccine development for Chlamydia trachomatis. The title of her Discovery Day presentation is From Human Immunity to Vaccine Strategy, Defining Protection Against Chlamydia Trachomatis. Please join me in welcoming Dr. Darville back to Pittsburgh. Toni.
Well, thanks for that wonderful introduction. I need a copy of that so I can send it to the NIH for my next grant review. It's absolutely wonderful to be here today. We were in Pittsburgh for almost 7 years. I was here when we moved from Oakland to this Taj Mahal of a children's hospital, and enjoyed lots of rounds in the hospital and lots of time in the beautiful Rangos Research Center. And it's absolutely wonderful to be back. It's a beautiful city with lots of good friends. We're staying through Saturday evening so we can enjoy some time with friends here. So the work I'm going to tell you about today is really started here in Pittsburgh. Because when I moved from Arkansas to Pittsburgh, I suddenly had the opportunity to do human studies, which I really didn't have that opportunity in Arkansas. We did all this mouse work. It was all on pathogenesis. But we moved here and I met Harold Weisenfeld, who was an OBGYN at McGee, and Sharon Hillier, who was a microbiologist at McGee. And both of them were really world-renowned physician scientists and scientists who studied sexually transmitted infections in women. And so Sharon and I met and we developed this proposal and were awarded a large grant to study human immune responses by recruitment of women through McGee Women's Hospital. And so that's where I'm going to tell you about the work that's come from that and how that's led us to a vaccine that we're now hoping we can move into human trials. So I'll start by describing the pathogen and the disease it causes, just to remind you what chlamydia does. It's not a flower. And then the problem space. Why is it that we still don't have a vaccine for this pathogen? And then discuss briefly the key components of immune protection and a vaccine antigen that we are using for vaccine development. Then talk about our approach to the vaccine development, highlighting what we learned from that approach, including both its successes and limitations. That sets the stage for our next generation platform in which we directly link the antigen to an immune stimulating molecule, a strategy that we have seen dramatically improves the protective T cell response. Finally, I'll close with the safety and translational implications of this work and discuss how this platform may inform vaccine development for other intracellular bacterial pathogens like TB. There's Lee. So chlamydia is an intracellular gram-negative bacterium that invades epithelial cells through multiple entry pathways, and that's important, rather than a single dominant receptor. Once inside, it replicates within a protective vacuole, co-opting host nutrients and shielding itself from immune detection. It infects multiple mucosal sites, including the genital tract, the gastrointestinal tract, the conjunctiva, and the respiratory tract of newborns. Although severe outcomes are uncommon at the individual level, the global burden of chlamydia infection means the cumulative impact is substantial. Antibiotic treatment is highly effective, but when infections go undetected, approximately 10% of women develop pelvic inflammatory disease, which may be symptomatic or subclinical and silent. PID can lead to chronic pelvic pain, infertility, ectopic pregnancy, and adverse pregnancy outcomes. Tubal scarring and post-obstructive hydrosalpinx of the oviduct underlie much of this risk, contributing to both reproductive morbidity and pregnancy-related mortality. Chlamydia is also the leading infectious cause of preventable blindness worldwide. Trachoma affects more than 20 million people, with over 1 million already irreversibly blind. Despite more than a decade of mass drug administration, transmission remains high in endemic regions, particularly among children, and the global economic burden exceeds $8 billion annually. We model infection using Chlamydia miraderum, which is host-specific for mice, and it recapitulates key features of human disease. This model captures ascending infection, epithelial cell tropism, and the development of upper tract pathology, including hydrosalpinx. The images here illustrate the close parallels between human fallopian tube damage and oviduct pathology observed following murine infection. But before moving into our vaccine data, I just want to briefly orient everyone to major CD4 T cell subsets, since these distinctions are important for the talk. When naive CD4 T cells encounter antigen with antigen-presenting cells, depending on the cytokine milieu that's present, they develop specialized functional programs. For example, Th1 cells produce interferon gamma and are central to cell-mediated immunity, particularly against intracellular pathogens like chlamydia. Th2 cells are more closely linked to antibody responses and defense against extracellular parasites. T helper 17 cells are very important in mucosal defense, and they help to recruit neutrophils for barrier defense. Then there's T follicular helper cells, which drive high affinity antibody production. And then, of course, regulatory T cells, which are the brake, and are very important, especially in chronic infections, to put a brake on the immune system and prevent chronic pathology. So years ago, using genetically deficient mice, investigators found that CD4 T cells are required for clearance of protection in mice. On the left, mice lacking MHC class 1 and therefore unable to generate CD8 T cell responses resolve infection fine, similar to wild-type mice. But when you look at class 2 deficient mice, which fail to stimulate CD4 T cells, you see that class 2 deficient mice never resolve infection. It goes on and on and on, and these mice have horrible disease, showing that CD4 T cells are really essential for resolution of infection. This next panel shows that the importance of interferon gamma. So if you look at interferon gamma wild-type mice, infection resolves within about 28 to 30 days. Upon challenge, mice are somewhat resistant to reinfection, so some natural immunity develops. But if you look at mice that are deficient in interferon gamma, they resolve a lot of their infection, most of it, but this low-grade chronic persistent infection continues on and on and on. And you can't really even challenge the mice without treating them with doxycycline because they remain infected. When you look at what happens to the genital tract of these mice, it's horrible. There's neutrophils everywhere. So what happens in the absence of interferon gamma is this huge T helper 17 response comes in and brings in neutrophils, and the genital tracts are just really full of purulent neutrophil exudate. So these data taken together show, again, the importance of CD4 T cell production of interferon gamma and clearance of infection. Well, what about B cells and antibody? Well, in this study where we looked at B cell, Morris et al. Looked at B cell deficient mice, you can see that they resolve infection similar to wild-type mice. So B cells are completely dispensable for clearance to primary infection. They do, and they resolve infection normally. When you challenge these mice, there is some reduction in the natural immunity that develops. So they're compromised in protection from secondary infection. But if you notice, these mice are still resolving infection by day 15, and the bacterial burden is 3 logs lower than it was during primary infection. To the extent that there is an effect, it is unlikely to reflect loss of antibody alone. It may instead relate to the absence of B cell antigen presenting function, which is important, or disruption of mucosal lymphoid architecture that helps support resident memory T cells. But taken together, these studies reinforce the central conclusion in mice that CD4 Th1 immunity is the dominant protective mechanism. Well, we moved to Pittsburgh, and there's 9,000 babies a year delivered at Magee Women's Hospital, and all the Pittsburgh women seek care there. And so fortunately, we paired up with a group there and recruited now over 500 women into various studies, including these two, which we call the T cell response against chlamydia cohorts. So these were young women who were at risk for chlamydia. A lot of them had already been diagnosed with chlamydia, and they were recruited into the study, going to various health clinics, adolescent clinics, the Allegheny County Health Department. And so they were referred into our group, and they were from 17 to 40 years of age, and they had diagnostic testing for the various STIs, lots of sexual history questionnaires, clinical exam, and lots of samples were obtained, including uniquely an endometrial biopsy. And this is one of the cool things about the clinical core at Magee is that we had a set of nurses who were very skilled at doing endometrial biopsies. And so we were able to assess whether chlamydia was in the upper tract or the endometrium of these women at enrollment. They also drew serum and blood samples that we could use for immune studies, as well as cervical samples that we could use for immune studies. Then what was really cool was these nurses, they got the women's grandmother's phone number, the boyfriend's phone number, the second boyfriend's phone number, and they called them all, and they came back for multiple visits. So they had an 85% retention rate in this study. And these women were followed over a year. They came back at 1 month, 4 months, 8 months, and 12 months. All the samples, all the questionnaires happened again, except for the endometrial biopsy. So this allowed us to examine factors associated with risk of reinfection and factors associated with protection from reinfection. In the initial cohort, 65% of the women were infected at enrollment, and there was about half of them had endometrial infection, and we've now seen these similar numbers in track 2. Upon longitudinal analysis, repeat infection occurred in 35% of the women, but 65% of the women remained uninfected despite ongoing sexual activity and risk. So the first thing we did was look at antibody. So what about, in humans, antibody may not be really important in mice, but what about humans? So we did actually 3 separate studies using samples from these women. In the first study, we looked at MIF, microimmunofluorescence antibody titers, and this assay isn't even done anymore in the world, but it actually looks for antibody to the whole organism. And we did this in all 250 women, and what we found was that although high titers correlated with lower cervical burden, they did not protect the women from upper tract infection, ascension, and actually higher titers of antibody were associated with increased reinfection risk. So the women with higher titers of antibody actually were the ones that were more frequently follow-up positive. We then did ELISA using whole chlamydia and looked at IgG and IgA, not only in serum, but also in cervical secretions. And again, found that these antibodies were insufficient to prevent endometrial infection. And again, increased anti-chlamydia antibody was associated with enhanced risk of incident infection. Finally, we looked at individual proteins. So we paired up with a German cancer research group who were very interested in antibodies promoting ovarian cancer because chlamydia is linked to ovarian cancer. They had this really cool immunoproteome that expressed 130 chlamydial proteins, and we looked for antibody to all those proteins, individual proteins. We found lots of antibody, but none of them were associated with protection from reinfection. In fact, some antibodies to certain proteins, again, were associated with increased risk for incident infection. Well, just to remind you how antibodies, the primary mechanisms of antibody protection, it's been shown in mouse models that antibodies can help kill chlamydia through opsonophagocytosis. So the antibody binds the chlamydial EB, the elementary body, and if interferon gamma is present, it can enhance phagocytosis and killing. Well, neutralization is the other mechanism, primary mechanism. And so if you think about most of the vaccines that we use in children are antibodies to specific proteins. COVID vaccine, for example, antibody to the spike protein neutralizes the virus and prevents it from entering the cell. The problem with chlamydia is there's multiple proteins on the surface that enhances its uptake. So we've now looked at opsonophagocytosis, and as you can see here, that definitely there is a small inverse correlation with the amount of phagocytosis activity and burden, but phagocytosis activity doesn't distinguish women who have upper tract infection versus those that don't, nor does it distinguish women who are follow-up negative versus follow-up positive. Other groups have shown also that this phagocytosis activity doesn't correlate with protection. And a recent paper from an Alabama group showed that they actually did neutralization titers and found no association of neutralization antibody titers with protection from reinfection. So leading us to now know that high antibody titers to chlamydia may mark failed T-cell immunity and not protection. So basically, they're a marker of exposure. The more you're exposed, the more antibody you have. So if you take an individual and their genetic tendency is to have a weak Th1 or interferon gamma response, then those are the women who likely develop prolonged infection, repeated infection, they have increased bacterial burden, and this leads to high antibody titers to chlamydia. This also reflects high antigen exposure, which, although driving a humoral response, also drives tissue damage. So overall, antibody titers are, having antibodies is not a bad thing, but it's just a marker that you're not developing the correct immune response, which is a T-cell mediated response. So T-cell response against chlamydia, these are track cohorts. We definitely looked at T-cells. So the first study we did was a collaboration with a vaccine company called Genosha, And we did this really very difficult assay where we took dendritic cells, exposed them to E. coli expressing chlamydial antigens, and we looked at 275 chlamydial antigens. And what we found was that the recognition of chlamydial antigens was increased in frequency in women who were follow-up negative when you looked at CD4 T cell recognition. So women who were follow-up negative in our study had a higher frequency of CD4 T cell responses to chlamydial antigens. There was no such association with CD8 T cells. Then my postdoc, Casey Younts, did a really cool study where she used CyTOF using 40 different immune parameters and was able to probe these T cells deeply. And what she found was that when we looked at follow-up negative versus follow-up positive status, women who had a follow-up negative status had higher frequencies of Th1 cells, CD4 T cells making interferon gamma, but also T cells of the T helper 17 phenotype as well. And these cells T helper 17 cells are very plastic, and they can actually morphologically change into Th1 cells when they're at a site of infection. Interestingly, in parallel with our antibody data, women that were follow-up negative actually had lower frequencies of effector memory Th2 cells. So those who were follow-up positive, not resistant, had higher levels of antibody inducing T cells. So this, to summarize, when you think about HPV, another sexually transmitted infection, we have a wonderful vaccine for it. It generates antibody, neutralizing antibody to these viral particles, wipe out the ability of the virus to invade the cervical cells. That's not going to work with chlamydia. What we need are the induction of CD4 T cells that make interferon gamma. And we know how interferon gamma works in humans to kill chlamydia. It actually depletes an essential nutrient for chlamydia called tryptophan. Interferon gamma induces indoleamine dioxygenase, which turns tryptophan into kynurenine that the chlamydia can't eat anymore. So chlamydia is starved. So this is the mechanism that we know now that needs to be induced for chlamydia. So both murine and human data indicate that CD4 Th1 cells are needed, but what antigen should we use? And what antigen could reliably drive that response? Well, using cervical samples from these women recruited in Pittsburgh, we looked just by RNA-seq at what chlamydia genes were being or most obvious in their cervix. And we found a lot of outer membrane proteins, which was not surprising given you have to have those outer membrane proteins for the chlamydia to survive. We also found this secreted protein called CPAF, or chlamydia protease activity factor, was actually very obvious in 12 out of 14 samples. And then we went back to our antibody data where we had looked at those 130 chlamydial antigens, and I've mentioned that we found a lot of antibody, but it wasn't protective. But we used these data to find which proteins of chlamydia or immunodominant are recognized by the immune system. And lo and behold, the most commonly recognized antigen was, again, this CPAF antigen. 75% of women had antibody to chlamydia, to CPAF. And of those who had antibody, there was a lot of antibody to this protein, much more so than the major outer membrane protein or MOMP or another outer membrane protein called OMCB. So again, pointing to CPAF being perhaps a key antigen that we could use as a vaccine target. We also looked at the T cell responses from women, and this work we did in collaboration with Nilu Ginatiliko, who's a phenomenal T cell immunologist from Oxford at UNC. And so we generated short-term T cell lines. We exposed them to now over 35 chlamydial antigens. And again, CPAF rears its head. And so it's recognized by over 50% of women in our cohort. We've now looked at men, and it's also the most immunodominant in men as well. And when you look at what are the responses, T cell responses to this protein, you see that it's a CD4 dominant response. So here's interferon gamma. So you can see that when we stimulate these cells in vitro, they make a lot of interferon gamma, and all the cells that are making that are CD4 T cells. So we use a mouse model for our studies. We wanted to know if our mouse model was good to study CPAF as a potential vaccine antigen. So we took mice, we infected them with a an attenuated strain of chlamydia, which is actually a wonderful vaccine for mice, and then a virulent strain of chlamydia. And then we looked to see what antigens do the mice respond to. And we found, again, CPAF was highly dominant as an immunodominant antigen in mice, much more so than OMCB or the major outer-membrane protein. And again, natural infection in mice drives this very CD4 T cell dominant response with no CD8 T cells being found that recognize this antigen. And perhaps more importantly, when we looked actually in the genital tract, we see lots of polyfunctional CD4 T cells recognizing CPAF in the genital tract after infection. So what is CPAF? Well, CPAF, chlamydial protease-like activity factor, is a very important virulence protein for chlamydia. It actually suppresses the oxidative burst and interferes with chemical-mediated activation of neutrophils. It cleaves off the 4-mel peptide receptor 2 and basically paralyzes neutrophils. So they can't release their nets, they can't make reactive oxygen species, and it also promotes release of infectious progeny through degradation of host proteins. It's a very powerful serine protease. It even suppresses expression of CXCL10, which is a Th1-inducing chemokine. And so in that way, inhibiting the immune response. If you knock CPAF out of chlamydia and put it in a mouse, it doesn't do as well. And that was done by Karthika Rajiv. So CPAF is highly conserved, meaning that it will also work for trachoma. It's secreted. It's immunodominant in humans and mice. So if CPAF is the right antigen, the next challenge was, well, what formulation are we going to use to drive a strong Th1 response? So here we turned to some friends at Vaxcyte, a California vaccine company, and there's some chemists there, and we were introduced to them by the NIH, and they had this really cool cell-free transcription translation system where they can feed the DNA in for a certain protein and make it in a vial, so there's a lot less LPS contamination. And the main thing about this is you can generate a lot of antigen very quickly and make it scalable. And so this is what we're using. So we took the CPAF antigen generated by Vaxcyte, and then we're like, well, we need to pair it with some Th1 inducing adjuvants. So CpG was one that we first looked at. It's in a licensed hepatitis B vaccine. CpG activates dendritic cells and B cells through innate receptor TLR9 and driving strong IL-12 responses and Th1 responses. We also looked at STING agonists. STING agonists are like cyclic di-AMP. They've been used a lot in cancer trials to try to rev up the T cell response. They've not done great in cancer trials, but they're now being examined in vaccine adjuvants and showing promising results. And it also activates a Th1 response. And then there's a nanoemulsion adjuvant called ASO3, which was used in the pandemic H1N1 vaccines. It showed a lot of dose sparing in those vaccine trials and during the vaccine efforts. And it works by activating local innate immunity, recruiting antigen presenting cells, and providing T cell help. So led by Dr. Taylor Poston in my lab, we evaluated these Th1 inducing adjuvants alone and in combination with CPAF, and we delivered them in this experiment intranasally because we thought we should engage the mucosal associated lymphoid system so that to enable programming of T cells to traffic to the genital tract. So mice received prime and boost vaccinations 30 days apart and were assessed for immunogenicity with antibody and T cell ELISpots, or we challenged them after vaccination and then measure their bacterial burden. So intranasal prime boost vaccination with CPAF admixed with a STING agonist ADUS100 provided by Invivogen induced modest CPAF-specific antibodies, including Th1 associated isotypes, but there were robust increases in interferon gamma producing T cells in the spleen in all of the groups where the STING agonist was present. So in every group where CDA or ADUS100 was present, we see these nice interferon gamma responses. And none of these mice had any adverse effects in their lungs or at all. So When we looked at the T cell response of these mice, we found that if ADUS 100 was present, we saw these CD4 dominant responses to CPAF that consisted of interferon gamma and TNF alpha, or TNF alpha and IL-17. And when we looked at the bacterial burden of challenged mice, you can see that, again, any vaccine that contains the CDA sting agonist, the green group is the one that's only CDA alone, induced about a 1.5 log reduction in bacterial burden over the course of the infection. And then when we looked at pathology, we can see that hydrosalpinx developed in 9 of 20 PBS-treated control mice, where 17 of 20 mice vaccinated with unadjuvanted CPAF developed hydrosalpinx. In contrast, the greatest protection from pathology was observed in mice vaccinated with CPAF and the sting agonist, with reduced frequencies of hydrosalpinx also seen in other formulations where sting was present. Consistent with these gross findings, histologic analysis showed that the sting adjuvanted CPAF vaccines produced the lowest oviduct dilatation scores, with CPAF plus CDA alone leading to a significant reduction in oviduct dilatation compared to unadjuvanted CPAF. So maybe the sting agonist was important. So just to summarize this first platform, we found that intranasal delivery was safe, that the sting agonist plus CPAF with or without CpG and ASO3 caused no lung inflammation. They induced low antibodies, but as we've found, antibodies aren't likely to be hugely important. It drove CD4 dominant immunity with interferon gamma producing CD4 T cells, and the sting agonist was essential for this to happen. Protective efficacy was about 1.5 log reduction in burden, but pathology was not, that target wasn't really met in our book. Hydrosalpinx wasn't significantly prevented, but trends toward oviduct reduction dilatation were observed. So these results prompted us to rethink our adjuvant delivery and think, how might we increase immunogenicity further? Well, there are limitations of soluble sting agonists. They have limited membrane permeability, they're rapidly cleared from an injection site, and they have inefficient accumulation in draining lymph nodes. And this is what was found in the cancer trials. They actually were shooting huge amounts of sting directly into liver tumors. To overcome these limitations, we turned to a strategy that has emerged. Direct conjugation of antigen to a pattern recognition receptor agonist, ensuring co-delivery to the antigen-presenting cells. So we went back to Vaxcyte, and Taylor and the chemists there, we used AlphaFold and all of the new technologies available to try to determine where we could actually stick this sting agonist directly onto our antigen. And so about a year later, this took a lot of iterations. We were able to generate CPAF with a DIBCO adder, and it has a spacer in it with a cute little cathepsin B cleavage linker. So this way, there's guaranteed delivery, co-delivery of our antigen and the agonist to antigen-presenting cells. And this cleavage site allows the sting agonist to float away out of the endosome and stimulate sting. And the CPAF can then be digested and presented on class 2 and presented to T cells. So they are generating this now in buckets, like a brewery. I mean, it's really kind of cool watching them make liters of this stuff. So let's hope that it works. But anyway, so we're like, well, the vaccine company was like, intranasal, really? I mean, Toni, come on, that's really going to be hard to get it through the FDA. So we're trying intramuscular first. We're now also looking at other routes, but trying to get past the mucus, past the cilia, the upper respiratory tract, it's a lot. And so drug companies, and that's the thing that you learn as a basic scientist and becoming a translational scientist and interacting with companies, is that this has to be scalable. It has to be manufacturable. We have to generate 1 billion doses of this stuff, and we need to shoot it into everybody. So let's make it as easy as possible. Okay, so we now have this CPAF sting conjugate, and we intramuscularly administered it to mice, and you can see that when we conjugated this sting agonist to our vaccine CPAF antigen, we see huge increase in numbers of interferon gamma producing T cells. So this goal was met. What's really cool here is that there's an 82-fold lower sting agonist dose than when you just admix it. And of course, the vaccine company is thrilled because these things aren't cheap. So this makes it cheaper. And again, the response is CD4 T cell dominant. The CD8 T cells don't care, but we get great polyfunctional cells with this conjugate vaccine. When we've now vaccinated mice intranasally as well as intramuscularly with this, just the conjugate alone. And you can see that either route actually led to a 2-log reduction in burden over time. In this experiment, we did see some reduction in hydrosalpinx, but not hugely statistically significant. But there's only 5 mice, so more of these studies are ongoing in multiple strains of mice as well. So QS21 came out, and QS21 is a really cool adjuvant that it works in a different mechanism. It activates inflammasomes. It's from the soapbark tree, and it's in multiple vaccines that we use. It's in the Shingrix vaccine. Who's had the Shingrix vaccine? I have. It's in a malaria vaccine called Mosquirix, and it's in an RSV vaccine. And when you go to the pharmacy to get your Shingrix vaccine, it comes in 2 vials, and it's mixed up in the pharmacy. So one vial contains the antigen, the other vial contains QS21 and liposomes. Pharmacist mixes it up, puts it in your arm. This is how it works. So we were like, well, if we're not really meeting the goal of protection pathology, maybe we even need more immunogenicity, right? So maybe we take our conjugate vaccine, we add it to QS21, just like these vaccines, and we'll get a better response. So that's what we've done. So we've taken our conjugate vaccine, we've admixed it with QS21 liposomes, and we put this into human macrophages for human translation. We definitely see significant increases in cytokine production, interferon beta, which we need for this vaccine to work, TNF alpha, and inflammasome-induced cytokines, IL-18 and IL-1 beta. When we put this into mice, we see significant increases in their antibody production with the addition of QS21 over the conjugate alone, with lots of Th1 isotype antibodies being produced. And then finally, looking at the T cell immunity, we see a 2 to 3-fold increase in interferon gamma producing T cells when the conjugate is added to QS21 with a CD4 T cell dominant response. And again, despite these big increases in immunogenicity, we're not seeing any adverse effects in general or at the site of IM or with intranasal administration. So IM conjugate induced 10-fold higher CPAF-specific interferon gamma T cells compared to the admixture using an 82-fold lower sting dose. It confers robust control regardless of immunization route. A 1.5 log reduction in burden is really pretty significant using this highly virulent mouse model of chlamydia. Co-administration with the QS21 adjuvant induced synergistic innate cytokine production in human macrophages and the strongest adaptive immunity that we've found with 1 to 2 log higher antibody titers, a 2 to 3-fold increase in polyfunctional T cells, and no adverse effects. In challenge studies, we're going to have these in May. Next month, I'll have the outcome of the challenge studies using this combination. So I think this platform actually also has broader implications for other bacterial vaccines. The conjugation generates a dose-sparing, scalable, and mechanistically targeted vaccine strategy. Co-delivery of the antigen and the innate stimulus reliably drives a CD4-Th1 response. So if your pathogen needs a CD4-Th1 response, the sting agonist is really doing that job. QS21 can amplify the responses through activation of the inflammasome, but the conjugate alone was highly immunogenic and well tolerated, supporting its potential as a simplified, translatable Th1 biased platform. So this work has addressed how to induce protective CD4-Th1 immunity when classical adjuvants fell short, aiming to establish a broadly applicable vaccine strategy for intracellular bacteria. We're actually exploring ways to actually conjugate sting to other bacterial pathogen proteins as we speak. So the T-cell roadmap to chlamydia vaccine success, rah, rah, let's hope. Human and mouse data have converged, right? We studied for years how disease happens, and then we really found out that protective immunity against chlamydia requires CD4 Th1 cells. We've built a vaccine platform that reproducibly induces that response, CPAP with a STING conjugate or lone or with QS21, and we are committed to a rigorous path forward. We are confirming safety and immunogenicity in non-human primates with collaborators at UC Davis. Again, also developed that collaboration while I was here, thanks to a Hartwell Foundation grant that I received at Pittsburgh. And called Dallas up, the vet there, she's putting these things in monkeys. We are then hopefully ready to ask the critical question, can we vaccinate young people at risk and prevent chlamydial infection? So NIH, drug companies are always talking to me, and at the end of my, immunology data, they all go, well, okay, well, if you're going to do a chlamydia trial, how would you do it? So I actually met with a clinical trialist. I am not a clinical, so I need to talk to Marion, to figure this out. But so back of an envelope, we kind of put some stuff together. So our proposed next step will be a multi-center randomized double-blind phase 2 study right here in Pittsburgh at McGee, enrolling healthy young adults 18 to 25 from a high incidence clinical site where baseline chlamydia positivity is approximately 15%. Now, the good thing about chlamydia is because so many people have it, it's not going to be real hard to find out if your vaccine works. So basically, 1-to-1, either our investigational vaccine, a control. The key objective is straightforward. Can vaccination reduce incident infection over a 12-month follow-up period? So participants would undergo serial surveillance, quantitative PCR, pre-specified time points. I mean, now with self-obtained lower vaginal swabs, you could get these girls to collect them every month if you wanted to. Of course, men would have to pee in a cup. So we would also, make sure what's their behavioral risk to account for exposure risk, and critically power this as a prevention trial. The primary endpoint would be incident PCR confirmed chlamydia infection within 12 months. This is a clinically meaningful endpoint. I mean, preventing acquisition of infection would be huge, but it could also be really difficult because this is a mucosal site. You've got to get the T cells there really quickly. So secondary endpoint is to look for bacterial burden. What we know from our human studies is that women who had upper tract infection almost always had over 10 to the 4th organisms in their cervix. So lower than that, the risk of upper tract infection was significantly lower. So no brainer, high burden means ascension, right? The more chlamydia you have in your cervix, the more likely it is to crawl up. So and lots of mathematical modeling studies, a couple have shown that if you can reduce the burden by 2 logs even, then you'll significantly reduce transmission to others. So if we can get people to have low burdens and we can vaccinate both genders, then we could actually, perhaps really prevent ongoing transmission of this disease and upper tract disease in women. So here's my acknowledgement slide. This is a picture of some of the key people that work on. These are my mouse group. They are like a mouse vaccine testing site, and it's wonderful. It's really fun. We're testing our vaccines at the same time we're testing drug company vaccines, and of course, may the best woman or man win. So best group won. So. Also, a shout out to Vaxite. They are absolutely phenomenal partners. They are friends of ours. They are chemists that will talk to us forever. And I've been able to use my old chemistry degree a little bit, which has been really fun. It took a while to get those synapses firing again. Eric Peruzzo is a French guy. If you ever want to go to France and see a really cool, meet a wonderful scientist, he gave us the STG 1151 even before our MDA was signed. Anyway, that's all, he's just a phenomenal scientist who wants to move the science forward. And of course, the wonderful people at Pittsburgh that really gave us these wonderful samples, and all the young women that, Ubered in and had an immunoneutral biopsy and then submitted blood and, talked about their sexual history. They're just phenomenal people, and the nurses that helped with that. And of course, NIH funding, none of this would have happened without NIH. And I have to show our newest lab member is Isla. This is my 5-year-old granddaughter, and she's 5 years old, going on 15. She's already talking about the boys in her class. Of course, they're older. They're transferred in from kindergarten. So, we definitely need this chlamydia vaccine. I mean, I've heard more about Kike than I ever want to hear. So Kike is really cute. I don't blame her. I don't blame her. But so yeah. I mean, I've got gray hair. I'm really, there's a limit to the time that we have to keep this doing. But I have a phenomenal team of young people. Some that I've brought from Pittsburgh. My lab director, who does all the mouse studies, came right here. She's a Pittsburgher. We have Steeler paraphernalia all of our lab. So it's wonderful. And so the hope is that we will see phase 1 trials or phase 2 trials before I'm done with this. Fingers crossed that this thing will work in monkeys, and then that we can move it into humans. So I'll stop there, and this has been really enjoyable. Thank you so much.